Tatum. I'm a doctoral student at San Diego State University in the Collusionia Lab, and today I'll be presenting on my first research project in this program titled Characterizing the Expression of Rhodopsin HMBR1 and Methylomicrobium alkalophilium 20Z, which I'll refer to as 20Z from this point forward. So the importance of this project is profound. In the Collusionia Lab, we study single carbon biocatalysis, and this project is a bioengineering project which combines microbiology and applied environmental science to create a strategy with the potential to combat greenhouse gases. So to begin with, I will give a brief overview of greenhouse gases and the importance to this study. So in this diagram, you can see we have our solar rays coming from out of our atmosphere into the Earth zone. And with the increase of greenhouse gases such as carbon dioxide, nitric oxide, and importantly methane, we have the rays coming into our atmosphere and becoming trapped within this zone, which increases our overall global temperature and global warming. So in order to combat that, we're utilizing Methylomicrobium alkalophilium 20Z, which is known as a methanotrope. Methanotropes are bacteria that utilize methane oxidation, and we're trying to use that organism also with a bacteriodopsin, HMBR1, as you can see here in this diagram, which is a light-activated proton pump. And the hypothesis of this study is if we're able to insert our HMBR1 bacteriodopsin into 20Z, can we increase the methane oxidizing potential of this organism? So with this study, we began with, again, the bioengineering, and I used Gibson cloning assembly to create a construct in which I was able to take our gene, HMBR1, and I can draw it up here, with the promoter and the terminator, and we have our HMBR1. And then with that, I used two different codon usage biases, 1 and 4. So for the purpose of the project, they would be obviously in two different constructs, but a codon usage bias is important because we need to optimize the efficiency of this bacteriodopsin in the organism in order to try to increase and create the best environment for methane oxidation and utilization. So this is the construct that we created with the codon usage bias, which is referred to CUB. We did CUB 1 and 4 for this project. So with the setup of the project, we began with having methane saturation and methane rate limiting experiments, two different sets, in which we began with creating a setup with our organism overnight in a flask with saturated methane, and then testing the methane concentration within that within our jar, within our semi-environment at periodic time points, and assessing the data analysis to see if this does in fact increase our methane oxidation of the organism. And then again, another setup with our rate limiting step with specific amounts of methane inserted into the environment, again to see how this affects the utilization of methane within this compound. So initially with our project, we were able to see that there is potential of this study, but the future direction is really to optimize our gene expression and potentially look at cellular cytotoxicity. This study is very important because it has the potential to really impact global warming and create a strategy to decrease those greenhouse gases which are toxic to human health and overall our environment. So future direction of this study is to come. Thank you.